So hello grade 12 students, so we'll continue with our exercises for chapter 5 and here we move to exercise number 4. Exercise number 4 talking about the karyotype. So karyotype, the said karyotype analysis allows the detection of genetic diseases. The next table, which table? This table, allows the detection of the genetic diseases karyotype analysis. This table shows the results of the analysis of some normal and abnormal karyotypes. Only the affected pair of the chromosomes are shown in this table. يعني بس affected pairs of the chromosomes. ما فرجوني كل الكاريوتايب، فرجوني only the affected pairs. So here, part A. At what stage of the cell cycle can we observe such chromosome sheet? The chromosomes, they are well seen and well observed at a specific stage of the cell cycle. This is known as, who can tell me which phase of the cell cycle? Hariri, which phase?
Metaphase.
Metaphase, very good. So بنشوف in the past they are described at the metaphase. What's special about the metaphase يا حسن وصفة? What is special about the metaphase? Chromosomes where?
At the
At the equator of the cell. They form what's known by equatorial plate. Very good. So part B, can we detect any abnormality or an abnormality before the baby's birth? فينا نعرف abnormality before the baby's birth? If yes or no. What do you think, Badia?
Yes, you can.
Yeah. Yes, by using what?
There's three tests we can do.
Three tests, and they have the special name. It's called prenatal diagnosis. Can you give me one of these names of these techniques for prenatal diagnosis?
Amniocentesis.
Very good. Amniocentesis is one of these prenatal diagnoses. So we can detect any abnormality before the baby's birth using the prenatal diagnosis. So documents 1 and 2 were obtained from the karyotypes of child C1 and C2. This is document 1. On document 1, here we have chromosome 14, chromosome pair 14, and chromosome pair number 21. And here we have document 2 showing the karyotype of the child C2, also showing chromosome 14 and chromosome number 21. So they said that documents 1 and 2, they were obtained from the karyotypes of two children, C1 and C2. So C1 in the document 1 and C2 in document 2, who show the same troubles in development. You have the same troubles in development. Part C, they said, bearing in mind that genetic makeup or the genes carried by the chromosomes 14 and 21 together is the same for C1 and C2, meaning that the karyotype of the child C1 and C2 regarding chromosome 14 and chromosome 21, they carry the same genetic information. They have the same genes carried by 14 and 21 together. So here they said, compare these documents to each other and to that in document 4. Document 4 is showing that karyotype of the father of the child of C2. This is the father of the child C2 and regarding chromosome 14 and chromosome number 21. First of all, let's start with document 1. Document 1, what do we have? Chromosome 14. This chromosome 14, they are homologous. We have two pairs of chromosome 14 that are homologous. While moving to chromosome 13, how many pairs do we have? We have 3 instead of 2. Okay? We have 3 instead of 2. While in document 2 of child 2, here we have 2 copies, but here we have also 2 copies. Here they are not homologous. One of these of chromosome 14 is longer or larger, if you want, than that in the other one, the other pair. So we have here a non-homologous pair of chromosome number 14 for the child C2. But the comparison with the child, with the document 4, father of child C2, father of the child C2, it has 2 homologous pairs of chromosome number 14 and as well as the same for chromosome number 21. So they are homologous, see, they are normal. First of all, if I ask you the question by raising your hands, please. What is the abnormality detected in child C1? Yes, I want to move to Hadil. What do you think? What is the problem in child C1 here? In this child, C1, what is the problem?
He has trisomy 21.
Excellent, under trisomy 21. While Muhammad, what is, thank you, Hadil. Moving to document 2, Muhammad, what is the problem? What do you think? If they have the same genetic information carried out, meaning that the same information carried by chromosome number 21 birth is the same as the one carried by chromosome 14. So what is the problem? What do you think, Muhammad? Yes, Muhammad? I think there is a problem in Muhammad's connection, I think. Muhammad, do you hear me? Okay, we can move to others. Let's move to Rama. What do you think the problem in child C2? Whenever Muhammad connections, it has problem or something. So, what is the problem here, Rama? What do you think in child C2? Yes, we have to compare it with the normal here. Like the comparison with the normal, Yarama. What's the problem? Uh-oh. Yes, Hassan?
One of the chromosomes
Yes.
longer than the other.
Okay, so في عندي problem شو بنسميها؟ Trans?
Can you hear me now?
Yes, I can hear you now, Muhammad. You had a problem with the microphone, I think, right?
Yes.
Okay, no problem. Yeah, Hassan. So you said one chromosome is longer than the other. So what's the problem here? What do we call this problem? It's called
Translocated.
translocation. So we have Robertsonian translocation, where one, since they have the same genetic information or the same genetic makeup, so this sentence is very important. So they have the same chromosome 21. So we have here another pair, but it was translocated to the chromosome 14. So this is what's known by Robertsonian translocation. Okay, moving now to what is the name of the abnormality the children they suffer from? We said that this is a trisomy, as Hadith said, and this is the Robertsonian translocation. Document 3 and 4 respectively obtained from the karyotypes of the mother. They said it's healthy carrier. And the father, which is healthy, of child C2. What did they say that it's a healthy carrier? Can you tell me why they said a healthy carrier? This is the question of part D. Why is the mother of a normal carrier? Why? Who can tell me? First of all, we have to compare it with the normal. This is the normal. This is our reference, if you want. Okay? So here we have chromosome number 14, and here we have chromosome number 21. But here we have translocation, so it was translocated to this. That's why it's called a mother, it's called a normal carrier. Part E, what are the possible gametes? We're going to go to the figure and we were going to estimate the risk. So part 1 or part A, the chromosome, they are well seen and observed during the metaphase. Can we detect the abnormality before birth? Yes. By what? By prenatal diagnosis, as Badia said. And we have different techniques, we can use them. Part C, the karyotype of the father is normal, so it shows 2 copies of chromosome 14. These 2 copies, they are homologous, as well as it shows 2 copies of chromosome 21, and they are homologous too. While karyotype of the child C1 show 2 copies of chromosome 14 that are homologous. Compared to the karyotype of the father, there are 3 separate copies of chromosome number 21, as Hadil said. So we have a trisomy 21, since we have an extra chromosome. Karyotype of the child C2, it shows 2 copies of the chromosome 14. They are non-homologous. One of them is longer than the other, while we have 2 separate copies of chromosome number 21. But bearing in mind that they said they have the same genetic information, child C1 and C2, so they have the same genetic makeup or the same genetic mass in both chromosomes 14 and 21. So the child C2 have the copies of chromosome number 21, where two of them they are separated, as shown here. Two of them they are separated, while the third one it was translocated to chromosome number 14. Yes, Badia, do you have any question? Raising your hand.
This one's from before.
Okay, no problem. So where two of them, two chromosomes, two pairs or two copies of the chromosome number 21, they are separated, and the third one is translocated to chromosome number 14. So child C2, they suffers from trisomy 21, Robertsonian translocation 21 to 14. Yes, Muhammad, your question is?
How is the mother a normal carrier?
Normal carrier, we're going to talk about this. Here, this is the question. I solved it. You didn't hear me before? We answered
No,
this
I
question. Okay, yeah. So karyotype of the mother, it shows two copies of chromosome number 14 that are non-homologous. One is longer than the other. And it shows one separate copy of chromosome number 21. So the other copy, sure, is going to be translocated to the chromosome number 14. So the mother has two copies of chromosome number 21. So there is no gain or loss of the genetic information. Two copies of chromosome number 21 and two copies of chromosome number 14, but we have translocation. That's why we call it a normal carrier. If you see here, it has two copies of chromosome number 14, and it shows two copies, but one of them is translocated to chromosome number 14. That's why we call it a normal carrier. Part 5 or part E, what are the possible gametes of the couple? Show the possible gametes. Here we have normal one, either it gives 14, 21. These are the possible gametes. It gives 100%. It gives one copy of chromosome 14 and one copy of chromosome number 21. But here we have translocation. Here either it gives 14 translocated with 21 and the 21 alone, or it maybe give only 14, or it gives 21 and 14, one of these, or it gives 14 and 21, which is the normal one. So we have four possibility, four different gametes. This one you have to draw it on your copybook. Here we have the translocation. Okay, so don't forget this. This is the translocated part. Okay, this is for the mother, and this is for the father, which is the normal one. Moving to the last part, what you have to do, you have to do factorial analysis. Doing the factorial analysis, we have the risk for trisomy 21, 25%, monosomy, it's 25%, normal carrier, it's 25%, and to be normal, it has a risk of 25%. Moving now to the last exercise, which is exercise number 7. Pay attention to this exercise. This is a very important exercise, and these are the exercises that are important for your quizzes and your exams in this chapter. Cystic fibrosis is a hereditary disease manifested by abnormality in the cell exchange, which lead to progressive blockage of the respiratory and digestive functions. Document 1, which is the pedigree of a family suffering from this disease. First of all, they ask the question, is the allele dominant or recessive? Justify your answer. So let's move now to Selena. What do you think? Is the allele dominant or recessive? Justify your answer. Looking to the pedigree, the blue one, they are the normal, and the brown or the orange, they are the abnormal or the affected one. Yes, Selena, what do you think? Is the allele dominant or recessive for the disease?
It's recessive
It's recessive. Justify.
because the parents are normal, but they gave birth to affected children.
Okay, you have to specify which parents in the pedigree. You have to use the pedigree, Selena.
Parents 1, 1, and 1, 2 are normal.
Okay, all of their descendants, they are normal also. So you can't take this. You have to take another pair, another couple.
Parents
this.
2, 3, and 2, 4.
2, 3, and 2, 4, they are normal, but they gave birth to?
Affected children 3, 9, and 3, 10.
Okay, so they are?
So the disease is recessive but was masked in the
The
parent.
allele of the disease was recessive and it was masked in the parent. Okay, then immediately whenever we prove dominant or recessive, we have to prove what?
Symbol.
Give the symbols. Yeah, let, for example, N capital be the symbol of the allele coding for the normal phenotype, and let's C small be the symbol of the allele coding for the cystic fibrosis disease. Okay, part 2. Yes, Let's move to Ali. Discuss logically the chromosomal localization of the gene responsible for the cystic fibrosis. You have to discuss, first of all, you have to deny it on the why. This is your job, Ali. Yes. Ali Hariri? Ali Hariri is not here. Okay, let's move to Muhammad. Deny it on the why. Also Muhammad, the connection went to Muhammad. I think so. So let's move to Rama. Rama, do you hear me? Rama?
Yes.
Okay, please. Can you deny it on the why? فينا ننفيها على why. This is your job. Okay.
Every affected daughter should have an affected father.
Every affected daughter should have an affected father. On the why. Pay attention. I'm talking about the why. If it's on the why, can we have females affected?
لا. Only
Okay.
males should be affected.
Yes, only males are affected, so it's not the case. So here we deny it on the why, right?
Right.
Okay, to the X, your job here, Rama. But ننفيها on the X chromosome. Can you?
Behind every affected daughter should have an
Yes.
affected
Every
father.
affected daughter should have an affected father. Here, allele of the disease is recessive. Let's take the 310. It's XC, XC, right?
Right.
And it's found on the X. She should inherit XC from each of her parents, 2, 3, and 2, 4. XC and XC. And here it's going to be XCY. And the father should be?
Affected.
So, but he is normal, so it's not the case. Here, we deny it on the X. نفيناها عن X, نفيناها على Y. يلا, next time it's for Muhammad here. Connection. Yes, Muhammad.
Yes, yes.
You have to deny it on X and Y, both. The homologous part of X and Y.
Yes, in case of recessive, you should look for an affected son and a daughter having a normal father.
Okay, go. Which parents, which
So-
daughters, and which members of the family? Yeah.
9 and 10 in
9 and 10.
generation 3.
Okay.
They have a normal father 3.
Okay. So first of all, if it's on the X and the Y, what should be the genotype of 9 and 10? First of all, this is a male, so it's XY.
XY,
And this is a female, XX. True?
XX. It's recessive, so only expressed in homozygosity.
So it will be XC, YC, and XC, XC, right?
Yes.
Okay, so here their family, there should be XY, and this should be XC, sorry, X. Let's move first with X. Let's put it X first before we move to the XC. X, X, right? So we have
Yes.
here X, X, okay? First of all, YC is going to be inherited from the father, which is 2, 3, okay? So it should be YC.
It would be YC.
And here we have XC. One of these XCs will be inherited from the mother and one from the father. So it's going to be XCYC, and the mother should be XC and XC. And both of them should be what?
Affected, but they are
Affected, but they are normal. So it's not the case.
The case.
And then the disease is going to be what?
normal.
Autosomal. So here, as you see, every time we solve the cystic fibrosis disease, from now on, you have to be familiar with this. Cystic fibrosis is going to be autosomal. Okay? تمام يا حسن?
Yes.
Okay, great. So they said, part C, Hassan, write the genotype of the parents 3 and 4 and their children. Justify the answer for each of these genotypes. So for example, if we have 2, they have the same genotype, we can work them as a 1. For example, 9 and 10, both of them, they are affected and the disease is autosomal, so we can work them together. But يهمك يا Hassan, give me the genotype of 9 and 10.
Since they are affected.
Okay,
so,
give me first the genotype before the justification. Give me the genotype first.
CC.
CC, both of them, they are CC. Okay, why, ya Hassan?
Since the affected allele is a recessive, and they
Yes.
are affected
And it should be expressed in what? In
homozygous.
homozygous. So genotype should be CC for both of them. Okay? Here you can work for 9 and 10 together. In order to save time, you can say 9 and 10 CC. Why? Because the allele of the disease is recessive and it should be expressed in the case of homozygous. Moving now to Noura. Let's try Noura. What do you think about 3 and 4? They are normal or affected? Noura, do you hear me?
Yeah.
Okay, can you try? 3 and 4, they are normal. Right? Or affected? What do you think?
I don't know.
3 and 4, they have the blue color. So blue color, they are normal or affected?
Normal.
They are normal. So you have to use the legend, Yanura, okay? Thank you.
Yeah.
Okay, let's move now to, you want to shoot Dana. Dana is here, or she went again? Okay, she went. So Hadil, what do you think? 3 and 4, normal or affected? This is first. Normal or affected?
Normal.
Normal. So first of all, they have the allele N, since the normal is dominant over the recessive allele for the disease. And the big question is, they are an N or NC?
And C,
NC. Why?
because they have affected children.
Because they have affected children. So they have the children, they have CC. They should inherit C from each of their parents. So they should be heterozygous. Moving now to 8. Ebadia, what do you think about the 8? It's normal, right?
Yes.
Okay, so sure we have the allele
expressed-
N, right?
Yes.
Okay, so what is the genotype? An
Yes.
N or NC?
NC.
NC. Okay, so it's normal, so it should be NC.
Yes.
What do you think, Selena?
C.
It's NC?
It could be NC or NN.
Yes, sure. So it could be NC or an N. We don't have a clear justification about this. Either it's an N or an NC. Why? Because it's normal, and the normal can be expressed in both homozygous and heterozygous. That's why we can determine whether it's an N or NC. Okay, Badia?
Okay, we can write this in the quiz in the them or
Yes,
both
between
يعني.
N or NC, unless we are going to see in document 2 what will happen. Okay?
Okay.
Okay, so let's move now to the researchers associate cystic fibrosis to a modification in the structure of a single gene located in a pair of a human chromosome. So a technique of chromosome fractioning permits us to obtain fragments of DNA that can be separated by migration on a gel. Okay, this document 2, and this can be identified, by labeling. Document 2, which is this document, can show the result of the migration of DNA fragments. They are termed A1 and A2. They are issued from the chromosomes of different members. They are presented in document 1. Part D, they said, link each of the fragments, A1 and A2. Here we have direction of the migration of DNA fragments. Here we have fragment A1 and A2, 2 fragments. And here we have the tested individuals, 3, 4, 8, 9, 10, 11. What are they? They are related to the pedigree. أنا مش جايبهم من عند المembers 2, 3, 4, 8, 9, 10, 11. They are related to the pedigree. The 3, 4, 8, 9, 10, and the 11. Okay? They said link each of the fragments to its corresponding allele with reference to the phenotypes revealed in the pedigree. First of all, I have to find out the allele of the normal and the allele of the abnormal. Which of the members in the pedigree and shown in document 2 that are affected by the disease? Yes, Rama, what do you think? Which members that are affected and they are shown in the document 2?
9 and 10.
9 and 10, excellent. 9 and 10, they are affected. Both of them, they are affected, and they show thick band or a normal band?
Thick band.
Thick band. This means that we have two copies of this fragment. This means that 9 it's CC and 10 it's CC also affected. So here fragment A2 it goes for the one for the disease, the allele of the disease. While the fragment A1 is for the normal. So A2 is for the disease or it's for the affected allele, and A1 is for the normal. When we're detected, we detected from individuals 9 and 10. Okay? So here we can determine whether 3, 4, they are homozygous or heterozygous. What do you think, Muhammad? 3 and 4, homozygous or heterozygous?
Individuals 3 and 4,
Yes,
they are heterozygous.
heterozygous, having genotype N and C, since we have two bands, A1 and
Yes.
A2, as well as individual 11, also heterozygous. So here they are NC, and here we have NC, and here we have NC. Part E, they said discuss and write the genotype of the tested individuals. Already we did this, which are presented. Compare them, these genotypes, to those of individual 3 and 4 and their children, which are obtained. خلصنا 3 and 4, 9 and 10 and 11. We still have 8. 8 يا بديع. We said that 8, it can be either an N, as Selena said, or an C, right?
Yes.
Okay, انت معي بديع ما هيك؟
Yes.
Okay, we said it may be an N or an C. Which fragment قلنا for the normal هي ال A1 يعني ال A1 is for the N وال A2 is for the C. نيجي للindividual 8 بديع. It has one thick
Yes.
band
Indeed. Yes.
of N. So
Yes.
the genotype is going to be what?
Yes. NN.
An N. So it's not no more NC. So it's 100%
yes.
an
NN.
So there is no chance for the NC. Why? Because this technique, it helps us to detect the genotype exactly without any ambiguity, without any, for example, confusion. So this is advantage of this genetic labeling technique. So this part F, it allows the detection of the genotype, precise detection without any ambiguity. Okay? Okay,
Yes.
Badia? Okay, so let's move to the solution. So this is the 10 is disease, as Selena said, so she should inherit the allele coding for the disease from at least one of her parents. So the allele coding for the disease has mass in the parent, so it's recessive with respect to the allele coding for the normal. We have to symbolize, to let N capital be the symbol of the allele coding for the normal phenotype, and let C small be the symbol of the allele coding for cystic fibrosis. If the gene coding for disease is located in the non-homologous region of Y, as Rama did, no females should be affected, or none of the females should be affected, but 10 is affected, so it's not the case. If it's found in the non-homologous region of X, then F is disease, so her genotype should be XC and XC, and should inherit one of these XCs from the father 3, where the genotype is going to be XCY, so he's going to be affected, but the father is normal, so it's not the case. If the gene is located on the homologous region of X and Y, daughter and son affected, their parent, they are not affected, but here we have X and Y, 9 and 10, they are affected, so their genotype is going to be XCYC and XCXC. So they should inherit XC from each of their parent and the YC from the father and the other XC from the mother. So the mother and the father should be affected, but it's not the case. So it is autosomal. Part C, 9, 10, and 15, they have genotype CC, Y, because they are affected or they are diseased, and then the disease is recessive. They should be homozygous, and they should inherit allele C from each of their parents, or they look normal. So the individual 3, 4, and 11, they should be heterozygous, having genotype and C. 8, it has only an N or an C. Maybe homozygous or heterozygous, since it's normal and the allele can be expressed in both homozygous and heterozygous, since the allele of the disease is recessive. For D, this restriction map or the zemogram of individual 9 and 10, they show one thick band. Thick band meaning that they are homozygous, having two identical alleles, in this case CC, while band A2 correspond to the allele C, and band A1 correspond to the allele N, the normal. Part E, electrophoregram of the individual 3, 4, and 11 show two bands, A1 and 2. So their genotype is going to be NC, heterozygous. While individual 9 and 10, they show one thick band, so their genotype is CC. While individual 8, one thick band of A1. So 100% individual 8 is NN. So here all of the genotypes, they are similar to that we discussed it in the pedigree, except for individual 8. Individual 8, it has two chances, either an N or NC. This is from the pedigree. But using this technique, it's 100% N. Part F, what is the advantage of this technique? The advantage of this technique is to remove any ambiguity about the genotype of the individual with dominant phenotype. So it allows us to detect the genotype with precision. So 100% Badia is going to be N and not NC, the individual 8. So this is related to this exercise. This exercise is very important. This is, for example, the Lebanese first session 2001. Okay, so 2001. This is an important session, and this exercise is similar to the one you are going to have in your quiz for the next week. Okay.